Thank you everyone for being here for CytoDyn presentation. At CytoDyn, our lead product is leronlimab Pro 140. We are developing leronlimab for HIV and cancer. I will be making forward-looking statements. Biotechs are usually, when they have a stock price below $1, they call them high risk, high reward. I'm going to make a case today that we are low risk, high reward. The reason for that is we hit our primary endpoint. I mean, it's all about getting your phase 3 completed. We completed that. And we have done much more than that. So I believe it's low risk, high reward. We started developing this product for HIV. Our first phase 3 pivotal trial is the world's first self-injectable product for patients who could die if they don't have this product. HIV does not kill anymore. There are products that will be able to make this deadly disease a chronic disease. But not for this population. This population, patients that have 3 or 4 classes resistance, they have AIDS. So we're not talking about a population that's just a normal population. It's a population that are in great need of this product. So this is the first self-injectable antibody for that population. It's an unmet medical need population. We also have another phase 3. That one is not a pivotal trial. That one is going to need a pivotal trial after it's completed. The reason that trial was going on is because we have a lot of interest on this product for monotherapy. Gilead and ViiV are working on the next generation of products for HIV. ViiV developed two component product for HIV. All the HIV products are 3 component. If they don't have 3 component, they're not a regimen that can stop HIV from killing. We've developed a product with 2 components. They spent several $100 million. This trial is single component, 1 product. It's not even a synthetic drug. It's humanized monoclonal antibody. We have patients who were taking 4 or 5 pills a day, 3 pills a day, an exact time they had to take it, because if they miss it, they get resistance. Resistance is dangerous in the world of HIV. And they have gone 4 years, some of them 4.5 years, without any standard care pills. Just simple injectable sub-Q at home, once a week, 30 seconds, and they're done. So that's our HIV story. The next one is graft versus host disease. We were surprised when Pfizer came out and says our HIV product has indication in GVHD, leukemia patients who need bone marrow transplant. So we did an in vitro study, $10,000 cost of the study. FDA gave us phase 2, skipping all the development. That's how much FDA thinks of this product. That's why we have fast track designation. That's why we got $28 million worth of grant from NIH at the early time where this product was being developed. TNBC, triple-negative breast cancer. Now we're into cancer. World-renowned oncologists figured out that there is another use for the mechanism of action for HIV product that are CCR5 antagonists. We're going to get to that. That's our very important study. Colon cancer, phase 2. We're not in phase 2. We want to file an IND, initial new drug application, for FDA to give us another phase 2 for another indication. And prognostic tests, we'll talk about that. We'd like to get approval for that this year, hopefully. We're going to talk to FDA. All the studies have been done. So what's the mechanism of this product that has so many potential? Perhaps next Humira. If you look at the cell, HIV doesn't kill anybody if it doesn't enter the cell. It's just another flu. You get a flu, you're fine. If it doesn't enter the cell, our product stops the virus from going into the cell. Before the whole thing starts, it stops it. And here's a magnified picture of the membrane. There's two receptors, CD4, CCR5. Dr. Paul Madden discovered that HIV has to interact with this receptor and then this one to get in there. So he invented leronlimab, Pro-140. He discovered that. Dr. Richard Pestel, on the other hand, discovered that there is another use for this. And Dr. Pestel will address you about the other potential of this product. So at the early time, when this product was being developed, 12 years ago, FDA gave it fast track designation, stating that there are clear advantages of leronlimab versus current standard of care. We have a letter from FDA that says you got fast track designation, and these were the reasons they cited. $28 million from NIH was given to this product. I gave the last $6 million back to NIH because I thought that this product has a lot more potential than just going after substance abuse population. That was 5 years ago. Today, I'm proud to say that that was a good path we took. Here's our first combination therapy. 52 patients were there because this is unmet medical need population. Primary endpoint hit, p-value 0.003. 81% of the patients who some of them never could have suppressed viral load. Suppressed viral load means you don't transmit HIV to somebody else. Very crucial for other people, not just for the patient alone. 81% had suppressed viral load. We have a letter from a physician saying, my patient for 27 years didn't have suppressed viral load. Do not take this patient out of the Pro-140 at the end of the study. So we didn't. Pfizer had 45% for 48 weeks. We had 81%. Ibalizumab, last product, got approved 43% for 24 weeks. We never had any serious adverse event with this product for the last 670 patients that is related to Pro-140 leronlimab. We don't. This product is safe. This product is really state about. So 40 patients from this population is still went to the extension. We still have close to 30 patients in extension. Some are gone for 3 years, close to 3 years now. 3 years. Regulatory pathway, we met with FDA. We are carefully crafting a press release to come out about our meeting with FDA just last December, just last month. We had a conference call with FDA to fine-tune our BLA, Biologic License Application. I'm very excited about that, and we will be looking at FDA minutes to just make sure that we reflect exactly what we got. We are this far away from approval, so we want to make sure that we indicate to everybody what's happening these last few weeks or months before we get to the submission of BLA. Monotherapy, we also discussed that with FDA. We will be putting out press release about that also. Our responders rate in the past was said to be 90% with 700 milligrams. We will be updating everybody about that. But we have a path to approval now. We got the higher responders. We set out so many things to do, done every one of them. And we're successful in every one of them. Haven't failed in combination therapy, primary endpoint, haven't failed on finishing the trial with a great result, haven't failed to get monotherapy to the finish line, we haven't failed to find out other indication for this product. So to that, let's look at the market size for combination therapy. I'm going to go with this slide. We had a hard time pricing this product before we put lower price here. But then, as we are talking right now with Big Pharma, which we said to everybody, we're talking to Big Pharmas, we realized if we priced it for HIV one price and for cancer another one, it's going to be disaster getting this license for each indication. So what we did is we put a $70,000 price. The current product for HIV unmet medical need is selling for $118,000 per patient per year. Current antibody for cancer is selling for $70,000 per year per patient. So we'll go with 70, stay with that with all the indication that we might have. The market size for this combination therapy is pretty significant, and for monotherapy is much higher. This is a report from independent report BioVid going to the patients, to the doctors, to payers, and they said that one of the things that they said is 95% of HIV patients said they would have no problem using this injectable. They're not going to worry. Some big pharmas says, hey, they might not take injectable. Big pharmas. We said no. Here it is. 85% take it, no problem. For monotherapy, 95% for combination. So that concludes everything about HIV. We were shocked when Pfizer comes back and says this product has their product, their CCR5 antagonist, which got approved, Maraviroc, has a black box warning, has side effect, has to be taken by patients twice a day pills. This is not that. So that product is not comparable. Ours, we did GVHT. We compare ourselves to Maraviroc in an in vitro study. We had good results. So we went ahead and sent that results to the FDA. We got phase 2. When I asked for orphan drug designation, the FDA says take 2 groups of 32 mice, inject all of them with human bone marrow. If all of them died, then that means human bone marrow kills the mice. But give half of them leronlimab. If some of them stay alive, you got yourself orphan drug designation. 100% of the mice stayed alive. 100% of the other mice died. Because it's 100%, we did it again and again and again. And the results same. The mice that had no leronlimab, their weight loss was drastic, night and day. So we got orphan drug designation. Then that's when we got shocked when we hear Dr. Richard Pastel, 500-page paper published in peer-reviewed journal. I published 1 paper. I got my PhD in mechanical engineering. He did 500, so figure it out. He comes to us and says, I discovered something. I discovered and I published it that the metastasis of cancer that is responsible for killing mankind can be stopped by your product. Now, when you find something like he did, you can't do anything with that. You got to have a product that does that. So Pfizer and Merck approached him. They did the study. He holds the patent. Now we hold it because we acquired his company. But then he didn't like Pfizer and Merck. He come to us. He said, yours doesn't have any serious adverse event or toxicity that is comparable to Maraviroc. It's hardly any toxicity or side effect. So I want your product. We said, we don't have any money. He said, we don't want money. We want shares of your stock. That's a valuable commodity. And we did the deal. We said, sure. So we have the brain trust in our company now. He talks about his discovery that needs our product. So we have patented everything. And it's been utmost honor for me to work with this gentleman. And I'm very pleased to introduce him so he can explain everything. Dr. Pastel.
Thank you, Nada.
Thank you so much.
Well, good afternoon, everybody. Can you hear me up the back? I'm very pleased to be here today and to be involved with CytoDyn at this pivotal moment in their history. I'm relatively new to the company compared to most everybody in this room. We formally sold our company at the end of November 2018, and a lot has happened in the last couple of months. I thought I'd share with you a couple of insights related to how and why the company has pivoted from HIV into cancer. In a nutshell, Nada shared with you that CytoDyn is a publicly traded company that is on the OTC as a market cap of around 155 million, that has successfully completed a phase 3 study for HIV, having met all its primary endpoints, that's currently conducting its BLA, and is a highly de-risked company in that context. My laboratory, about 7 or 8 years ago, had been investigating the mechanisms by which a cancer cell metastasizes. And at that time, it wasn't understood exactly how that happens. And it's the spread of cancer that kills people. Nada mentioned to you today that there's a receptor called CCR5, which is essential for entry of HIV into the T cell. That ain't what it usually does, okay? What CCR5 normally does on a T cell is to direct its movement. So when a T cell activates CCR5 or its ligand, it moves. So one of the normal functions of immune cells, they're policemen of the body, tracking down infections and inflammation. And the cell moves because of this CCR5 present on its cell surface. Now, we found that, surprisingly, when we used an artificial intelligence approach, that cancer cells turn on that same receptor, and when a cancer cell does that, it starts moving. So we showed that this receptor, normally expressed just on T cells, promoting migration to the bones and brains and the other place where immune cells go, does the same thing for a cancer cell. So the CCR5 is necessary and sufficient for the movement of cancer cells to the bone, to the brain, to the lungs and other tissues. Now, Nada had mentioned what we had done, which was to study a variety of different cancers. And I'm going to show you some primary data, but just to preempt it, we looked at 2,200 patients with breast cancer. We looked at every gene in their cancer, and we found that about 50% of breast cancers overexpress CCR5. 50%. So the previous blockbuster, which was Herceptin, targets about 12 to 15% of human breast cancers. But unlike Herceptin, CCR5 is turned on in other cancers. It's turned on about 50% of prostate cancers, pancreatic cancer, colon cancer, a variety of different types of metastases. I was particularly struck by colon cancer. Both my parents died of metastatic colon cancer, and my father was a cancer surgeon. He was here at Sloan. He was at Sloan Kettering, and ultimately moved to Australia, to Perth. Big guy, 6'2", high jump champion, sprint champion, and when he had metastatic colon cancer, he couldn't stand up to take a pee. And so this is a disease that brings the strongest man or woman to their knees. So when we found that there was a receptor which was targetable, that was driving that metastatic process, I was very excited at a very deep level about the potential of shifting this game in the cancer metastasis field.
There's no session after you, so if you want to go a bit longer, you're welcome to do that.
Thank you. Excellent. So this is some of the primary data, and this is a study with 2,200 patients, so patient 1, 2, 3, 4 to 2,200. And this is every gene in their cancer. And this arrow means this is CCR5, and so CCR5 is turned on where it's red. So all these patients have CCR5 turned on their cancer, as do these other patients. But I was very focused on this group of patients because triple-negative breast cancer is a terrible, terrible disease. Now, I've been looking after patients for nigh on 30 years, and metastatic triple-negative breast cancer typically kills a woman within 9 to 10 months of the time they appear in your office. It typically attacks Ashkenazi Jewish women, women of that descent, African American women, and we currently don't have any targeted therapy. The standard of care is chemotherapy and surgery. So identifying this as a potential target, CCR5, in all these women was very, very exciting. And this is, I hope you can see this, these are the breast cancer cells here, and this is they're metastasizing. In this case, they're invading through matrigel, and this is their journey. And here you can see we add leronlimab, Pro-140, or we use this small molecule inhibitor. We dramatically cut that invasion. It's dose dependent. We up the dose. We block the invasion and metastasis. Now, NADA had intended to mean that when I had used the Merck and Pfizer drugs, I had demonstrated that they blocked metastases, but I was unhappy with those drugs because of their associated serious adverse events. So my prism looking at things is, how do I help my patients? You know, my training was in oncology, start off in bone marrow transplants. And when you add something in to current standard of care, you usually add it into chemotherapy, some sort of quite toxic treatment. So when I was doing the thought experiment of adding in a small molecule inhibitor to the standard of care chemotherapy, where the small molecule inhibitor had a lot of serious adverse events, I was concerned at how this might actually get into clinical practice. So as Nada mentioned, I contacted CytoDyn and asked if I could test their antibody in our assays. And this was the first result where we demonstrated that Pro-140 binds to cancer CCR5 and it blocks this migration of cancer cells. Now, if you're an immune cell and your CCR5 is activated and you start moving, the first thing you do is you turn on calcium signaling. That's the gas going into the car. And that's also true in the case of a cancer cell expressing CCR5. So we looked at calcium signaling as an initial surrogate endpoint. And this is calcium signaling here. This is a little blip, and we can measure this calcium signal. But when we add leronlimab or a small molecule inhibitor, we block that calcium signal. So basically, you block the gas going into the car and it doesn't move. So we next tested this in mice, and I'm going to show you two types of cancer. There are others. First, the breast cancer story. So these are mice, and these are the lungs of mice that are filling up with metastatic human breast cancer with time. So here we see 4 weeks, and the mouse lungs are now filled with this highly metastatic breast cancer. And these are the animals that have been pre-treated with a CCR5 inhibitor. And you can see this quantitated here. There's about a 95% reduction in metastatic burden. So these animals die and these animals live. And you can see the slope of this curve. So this is not putting on, a 10% improvement in survival. This is a 95% reduction in metastatic burden that ultimately kills the mouse. When we look in prostate cancer, we developed a new model of metastatic prostate cancer. Part of the reason we haven't really made a lot of impact in treating and reducing mortality from prostate cancer metastasis, we don't have great models to test it in. So we spent 4 or 5 years developing this model, and this is an immune competent mouse model. So they have a normal immune system. And these are the metastases of the mice. This animal's back is bent because it's broken. These are the metastases here in the spine of the mice. So highly metastatic prostate cancer spreads to the bones and brains of the mice, kills the animals. But the animals that were pre-treated with a CCR5 inhibitor blocks the metastases to the spine is not broken, and these animals are able to run around the cage. Now I'm going to fast forward to human studies. Suffice it to say that a lot of laboratories around the world have moved into this field. CCR5 has become the new cancer kid on the block. Very hot topic both in the research area and the laboratory, but also now in the clinic. This is one example of some studies that were conducted with CCR5 inhibitors. This was personally very exciting to me. This is a patient with metastatic colon cancer. And the way you see the metastases here, where it lights up red, just like in the animal studies, these are the metastases, these are the lungs of, these are the liver of the patient, and these are the metastases here. So every patient in this clinical study had failed all possible available therapies, and their disease was progressing. So that was the entry criterion into this study. And this is the result after 60 days of treatment with a small molecule inhibitor. The metastases dramatically reduced tumour volume. Now, this is in of itself a very exciting result, but more importantly, when Dirk Jager and his group, colleagues who work at the equivalent of the German Cancer Institute, the National Cancer Institute in Germany, when they looked at what was happening in the metastasis, there was an activation of the anti-tumour immune response. So in addition to the effect to block the GPS or spread of the cancer, it was also activating the policeman, the anti-tumour immune response, to go out and kill the metastases. So 2 independent belts and braces, 2 shots on goal, 2 different effects of the same CCR5 inhibitor. So based on these and other studies, we have decided to move initially into the area of triple-negative breast cancer, in part because of the current lack of options for these women, in part because the opportunity to impact more than 95% of those patients because they all express CCR5. The trial was approved by the FDA. We have moved forward, therefore, with this. The design is very straightforward. It's simply adding Pro-140 into the standard of care, carboplatin. The institutions that have signed up to be participants in this clinical trial are very high level. Northwestern University, Lurie Cancer Center, the Vanderbilt Cancer Center, Houston Methodist, Thomas Jefferson University, Sidney Kimmel Cancer Center, and others. Because of the enthusiasm, because this type of technology is without serious adverse events and has shown dramatic preclinical results. This is an example of colon cancer, and again, as Nada mentioned, there's about a 60 to 70% reduction in the tumour volume of colon cancer in mice. I want to spend just one moment mentioning that we are involved in non-dilutive funding efforts for the company, and that occurs now at the highest level. The strategy of the board and senior management are unified around the importance of forming strategic alliances to identify the shortest path to revenue in this company. Usually in cancer, the opportunities arise earlier in that pipeline, or usually successful outcomes in phase 2 are the results that lead to strategic alignments and new revenue coming into a company. We've also focused on licensing a technology for prostate cancer prognostic testing. In a nutshell, about 1 million men a year are diagnosed having serial elevated PSA, sent to their doctor, and the doctor says, well, we need to stick a needle into your prostate 12 times and we'll let you know what that shows. And after the prostate biopsy, about 250,000 men each year will have been diagnosed with prostate cancer. The question is it a prostate cancer that will kill you, or will you die with it from some other cause? And so this test distinguishes bad outcome from good outcome prostate cancer. There are currently 3 incumbent tests in the marketplace. They sell from between $3,500 and $5,500. And this test has shown in 3 independent clinical studies, 5 to 10 year follow-up, to be far superior to the incumbent technology. So we're pushing hard to ensure we have a 510K with the intent of licensing this technology. So I'd like to summarize where the company is at right now. It is an inflection point in the company's history as it pivots from HIV into cancer. The strategy around cancer is to initially provide proof of principle with triple negative breast cancer, and then really to look for a FDA approval for mechanism. So the intent is to move from triple negative breast cancer, CCR5 positive triple negative breast cancer, into CCR5 positive cancer. And this is a strategy that's proved successful for other companies due to the FDA ruling and the strong advocacy of the survivors, cancer survivors in this country, and a very wise move. So again, to recap, the BLA submission is moving forward under Dr. Paul Hassan's leadership, and is moving forward effectively with a revenue potential that in the first year is listed as being relatively modest market penetration considering the size of this market, which is now, you mentioned, about $3 billion potential annual revenue. There is a phase 3 pivotal trial which intends to be initiated. We have FDA approval for this triple negative breast cancer study, and we anticipate interim results within the first half of this year, as we anticipate results upon reinitiating the graft versus host disease study. We are seeking licensing of this prognostic test upon completing the 510K, and there is the intent to move into other cancers in 2019, starting with colon cancer, based on the successful outcome and the movement of Merck and Pfizer into this space. So the company has come a long way in the last 3 or 4 months, pivoting from historically an HIV immune-focused company, pivoting into cancer. Early results are very exciting in that domain, and I would agree with Nada that this is a company that has been de-risked on the HIV side and has extraordinary upside potential in the cancer domain. So I'll stop now, answer any questions, and thank you for your time.
Any questions? Go ahead, please.
Doctor, I didn't catch your name. I'm not sure you introduced yourself.
Richard Pestel.
Yes, Pestel, yes. Are you primarily working in clinical research at this point, or do you have a medical practice? How important is the CytoDyn relationship to you personally?
About 22 million shares.
Gets your attention, yeah.
Most of my time is spent on the road fundraising and overseeing the expansion as Chief Medical Officer into the cancer space. So I've been responsible for ensuring that we move rapidly and effectively into the cancer space, overseeing the construction of the IND for triple-negative breast cancer, identifying the relevant clinical sites, checking those sites out, making sure that this study is done effectively, and the responsibility here is to not fail the technology. The technology is beautiful. We just need to make sure that we do things exactly the right way and get the best people in the country participating. So the person who's doing the immunohistochemistry for CCR5 is the top guy in the country out of Minnesota. We've got the top guy looking at circulating tumour cells, the father of CTCs out of Northwestern. And then we've got 4 or 5 clinical sites that are some of the best in the country. So that's how I'm spending 98% of my waking time.
It's a very exciting story, I gotta tell you. Makes me want to go out and buy the stock.
Go ahead, you got my permission.
Join me.
Go ahead.
Can you talk about the monetization strategy for Pro-140 as it relates to HIV? I mean, would you sell that as an asset to someone like a Gilead or ViiV, or just license the technology? Because, I mean, a company involved in HIV might be a completely different kind of company involved in cancer.
Right. No, thanks for the question. The question is, would we be selling HIV product by itself, perhaps, and fund the rest of the program? We are in discussion with Big Pharma, but when you want to change Big Pharma's normal path of business that they have conducted for many years, making great amount of money and helping patients of the world, which is a great thing they have been doing. Now you're going to go there and say, okay, we're going to do it different. So that's going to take a little bit time to get that mentality to accept something like this. Would the results of our data convince them? We are in discussions, so we don't know anything how they're going to react, but we are in discussion. There has been interest, and we are exploring it, and we are definitely open to selling the HIV for the right price according to our third-party BioVid. That's the reason we did that report, to make sure we know what we're worth for them. Any other questions? Go ahead.
Can you give us some revenue guidance?
The revenue for the price that we have now, which is $70,000 per year per patient. If you look at 49,000 patients being impacted by that, you're talking about over $3 billion worth of market. There is a lot more research needs to go to that. But the bottom line is, we're talking about patients who don't have other options. They have got resistance. Now, there is another antibody that got approved, but that one is for patients have to go to hospital or a care center to get an IV every 2 weeks. Our advantages to that might be they can self-inject. That's a great product also. But they had a 43% suppression rate at the end of 24 weeks. Ours was 81%. The revenue that we are putting out here is based upon BioVid report, which was independent from us. They did a report. We believe monotherapy will also do a great job, but as you said, maybe we should license the HIV, because we are now looking at something much, much bigger than HIV. All cancers that we could compound, not all cancers that, all the cancers that we are allowed to, or we have potential for, we're going to explore all of that. So that's a huge market. So.
When will revenue start?
The revenue starts after we have our BLA completely accepted by FDA. We had put a timeline of March, which we have to now, based upon our discussion with FDA, we're going to have to put a press release out saying where is our timelines. When you give your final BLA, you got 10 months of standard waiting. We have accelerated approval. If that gives us 6 months, then you're talking about, so I'm confident in 2020 we'll have revenue. That's what I believe. Did you want to add something?
The only thing I was going to add is that Nada mentioned the pricing of around 118,000, and you have to go to a professional institution, either a hospital, to get an intravenous injection every 2 weeks. So there's an additional cost on top of that. So I think the price point of 70,000 may be a tad conservative, and many of us have thought pricing around 110,000 is entirely reasonable in this particular space. I think it's important to understand the company has been spending a lot of its money on producing commercial grade antibody which is ready for sale. So a lot of companies don't emphasize that, but this has been going on all the time behind the scenes.
And the importance of that, maybe, if I may add, is if you get a breakthrough designation for, let's just say, triple-negative breast cancer, FDA is not going to give you approval if you don't have commercial product. Approval for what? Because you have to still go get your commercial product, which will take 2 years. For the last 2 years, we didn't stop that. So we spend a lot of money, and everybody is saying, why are you spending so much money? They don't realize we spend $190 million so far for all of these things, and usually it takes $500 million to get a product to the market. So I think it's a very satisfying for me to say that we didn't use $500 million. We use this much, and we're where we are, and we do have commercial products sitting in the warehouse right now.
Yeah, so you spent, $190 million, and you got 200 million of commercial grade antibody sitting in a warehouse. I think, and you developed the technology for free, basically. It's pretty astonishing. Yeah.
Go ahead, please.
Gentlemen, thank you for an excellent presentation.
Thank you.
I know I certainly share your excitement. I think we have a decent amount of clinicians in the room. Is it possible to just walk us through top line your breast cancer and your GVHD study?
Sure. The design of the triple-negative breast cancer study is relatively straightforward. There's a dose escalation phase for 18 patients with the intent, we believe, of confirming that 700 milligrams is safe in this patient population. As you know, there's been some 660 patients treated to date and with leronlimab, and at this stage there have been no serious adverse events. But the reason for doing this dose escalation is we'd like to use the 700 milligram dose, and the subsequent 30 patients will be treated with that 700 milligram dose if there are no issues with that dose in this patient population. Important to this study is we do measure what's called circulating tumour cells. So although the primary endpoints are the standard endpoints as you see in cancer, progression-free survival, overall survival rate, we're also measuring a blood test. So we take blood from these patients 21 days after we give them leronlimab, and we measure the circulating tumour cell number. So this is a secondary endpoint, a surrogate secondary endpoint, and this information will be provided to the FDA as an earlier time point. So our intent is to use the circulating tumour cell data, share that with the FDA expeditiously. In terms of the graft versus host disease study, which we intend to reopen, the plan here is to change the entry criterion. That's the only significant change in the way in which the study is designed. And the intent here is to include only the low dose radiation priming group of patients, and to have a common conditioning regimen for all the patients. Before there was a lot of different conditioning regimens, and it's very hard to see a difference when everyone comes to you with a different background treatment. So my intent was to meet with all of the thought leaders, all the principal investigators, having met with them all over the last couple of months, get a consensus around what a common conditioning regimen would be, and the selection criterion has now been modified. So the intent would be to reopen that study. Again, this is a 14-week study in GVHD. So we're not talking about 2 years. This is a 14-week study. And I think it's important to emphasize, in the cancer space, the bar is set at quite a different level to HIV. 14-week study in GVHD. Please.
Before everybody leaves, I'd like to introduce myself as Tom Ross. I'm president of Palm Beach Business Group. We are hosting a breakfast for CytoDyn on Wednesday morning in West Palm Beach at the Embassy Suites near the airport, easy drive. And here's the information if anybody be interested in conceivably attending. I think we'll all agree it's a very exciting company. And this is an opportunity to learn a little bit more and so on.
Yep.
If I can go back to the HIV, if I recall correctly, when Gilead bought Pharmasset for the $11 billion in valuation, the Pharmasset asset was a lot earlier in the stage of the development and proof than where Pro 140 is. I mean, or said another way, Pro 140 is a lot farther along, almost near approval, than where Pharmasset's assets were. Is that correct?
So I'm going to let Dr. Pesta answer that, but I can only say one comment about that. Gilead passed on that opportunity few years ago for around $4 billion, end up paying $12 billion or something,
11 billion.
just 11 after that. But do you have any information on that?
So I think embedded in the question is what has been the interaction with Gilead at this stage and what is the level of understanding of what's going on here. And I think my impression is that there's not been the level of communication within the organisation as to, and there hasn't been the same sort of transparency as there should have been. And we're working on that aspect of communicating more clearly to the higher levels of the organisation now. Perhaps that's the best way to state that.
But they did buy Pharmasset,
Yep.
And they bought their assets not having been as proven as Pro 140 is actually proven.
Yep.
Yeah, exactly.
I agree. 100% agree with you.
Yeah, that's
Yeah.
a great point. Yeah, great point. Any other questions? Yeah, please.
Did you decide to use 700 milligrams for TNBC patient, Dr. Pesta?
Yes, so the study design is a dose escalation for the first 18 patients, 350, 525, and then the 700 milligram. And if we see no issues with dose-related toxicity, which we don't anticipate, then we'll use the 700 milligrams. We just wanted to go slow, do it safe, do it right by the patients. We don't anticipate there are going to be any adverse events related to the dose escalation component of the study, but we thought it was better to go slow, go safe.
And one thing I would like to add to that is that the first 18 patients that we do dose escalation, we're still taking the CTC, circulating tumor cell, for these patients also. So in the event, those first 10 of those 18 or 15 come out to have the CTC that's reduced drastically, the first thing we're going to do is to ask about breakthrough designation. We might not get to phase 2, stay in the dose escalation.
But your primary analysis of the first 18 is really a safety analysis.
Yes.
With efficacy.
Correct.
And as long as the safety piece is there, then you'll proceed to 700 milligrams.
Exactly. Yeah. Now, I think Nada's pointing out that there will be new information coming out relatively soon in several different high-impact areas, which we think will be very helpful for the current investors and the current round of investment.
And the study dose for the graft versus host disease, is that set at 700 milligrams for the 13 patients?
Yeah, rock solid. Yeah.
Okay. Thank you very much, everybody. I appreciate it. Thank you.